Hello, everyone, and welcome to our webinar dealing with data paralysis and pharmaceutical process development. My name is Jesse Harris, and I will be your host for today's presentation. I have two guests this morning, Mathew Mulhern from AbbVie Pharmaceuticals and Joe DiMartino from ACDLabs. Before we get into that, though, I want to let you know that you can submit questions during our presentation today. If you look within the GoToWebinar panel, there is a box for questions. It's on the right-hand side, so you can just drop any questions in there, and we will hopefully get to all of them at the end of the presentation. There will also be a recording made available, so for anybody who wants to rewatch the content or share it with your colleagues, you should expect to get that in your emails very near future. If you're having any issues with the presentation this morning in terms of audio or video issues, if you exit out of GoToWebinar and then rejoin the webinar, it'll let you back in, no problem. But that should resolve most issues. But if you have anything beyond that that isn't being fixed by that, just send us a question and we'll try and help you out as best we can. But with that out of the way, I would love to get started, and that is introducing Mathew Mulhern. He is a principal research scientist in AbbVie's Process Research and Development Organization. He joined AbbVie in 2001 and is as a discovery chemist before transferring to process chemistry in 2007. So I'm going to pass things over to him.
Hi, everyone. So as Jesse said, I'm Matt Mulhern from AbbVie Pharmaceuticals. I'm a principal research scientist. So let me give you some of my experiences, with working with these data-rich experiments and how this is leading to some data paralysis inside of pharmaceutical development. So the overall trend inside of pharmaceutical development is that we're running these data-rich experiments. We're bringing together a lot of different information from a lot of different sources, both online and offline. And what we're trying to do is make these correlations between the conditions used in these experiments, the parameters in these cases, and the results, whether that result is impurity data or maybe NMR data. But as we're generating these large data sets, we start to run into this problem. What did we actually learn? And how do we even interpret these large data sets? This data-rich experimentation can very easily become data paralyzing. We gather all this information, and then we try to think of a use for it later on. How did we actually get to this state? Well, just in a blink of an eye, we've gone from having mercury thermometers that you would stick inside of a reactor, and if you wanted to have data from that, you'd have to walk up to your fume hood, take a look inside, record that temperature into a laboratory notebook. Maybe it was even a paper notebook at the time. We've moved on to simple sensors. Maybe those were still being recorded then with a paper notebook through visual observation, or maybe it was tied into an electronic notebook in its first generation, which wasn't much more than glass over paper. But nowadays, we've moved on to things like, pictured here on the screen, these Mettler-Toledo reactors. They're gathering data from multiple different sensors and probes, and they're doing so every 2 seconds. So we start generating these huge data sets, but how can we still get it to something useful? So we need to capture that data from those reactors and still marry that to this offline data analysis, such as HPLC data. Current practice, we move to something like Excel or a spreadsheet, mostly because it's readily available. So this is an example here of just an Excel spreadsheet with all these different data points. So how do we get this to something that I can actually start diving into some details, but also get to something where it's manageable, something for visualization, because that's how we end up looking for our first round of information about the actual reaction itself. Counterintuitively, what we actually start doing in most cases is reducing the amount of data that we're working with. Maybe we were capturing data every 2 seconds throughout the reaction, but maybe we didn't need to be capturing at that data rate. So the first thing we're going to do is, as an example here, capture data that was only there every 1 minute. Perhaps we would even truncate this further to say, rather than looking at an entire reaction sequence, we're only looking at one portion of that overall reaction. The overall goal with this data reduction is to make the data set more manageable so that the processing on the computer in order to create our graphs and our visualizations doesn't take as long, and it's a little bit more interactive for us. But even after we've done this data reduction, we still have another part to our workflow, and that's bringing in our offline HPLC analysis, Could be any type of analysis really, but any offline result that we want to bring into these overall conditions that were captured from the Mettler-Toledo software. So in this case, I'm giving an example of some HPLC data. So we had the information about when the sample was taken, and then the impurity information that was generated. So now we finally made that correlation between our conditions and our results. So the last piece is doing the visualization. And this is where we can get into some of that analysis. We're looking for excursions in terms of some of the conditions that we're running. Maybe we had a temperature spike in the reaction. We can also start to look at those parameters and their impact on whether it's a starting material that's being consumed, products that's being formed, or maybe impurities as well. Were those impurities generated throughout the course of the reaction, or did they only start to form after the main reaction was complete? So that time course information is highly important, obviously, inside of the pharmaceutical industry. The other piece that we need to really focus on is the ability to transfer this information across scientists. Right now, most of our Chem ELNs don't do a very good job of letting us compare results from one scientist to the next. And that's where the beauty of Luminata really comes into play. It's that data accessibility, that ability to look at that single source of truth. And on top of that, we also have the ability to make use of the Spectris platform, and that involves Spectris Processor. Spectris Processor is able to read over 100 different data file formats. For us, that has a lot of importance because it doesn't matter then whether I use Agilent ChemStation software for my HPLC analysis or one of my colleagues uses Waters Empower system. In both cases, it's an LC system producing the same type of results. In the past, we couldn't bring those two results together except if we used something like a PDF or extracted data into some sort of a CSV and worked through a very complicated methodology. In both cases, with a Spectris platform, you convert these to a Spectris file, and then you can immediately go into Luminata with that type of data. We also can enforce a certain consistency of peak naming. If I call something dimer 1 and my colleague calls it dimer A, we suddenly have a disconnect. And inside of the Spectris platform, that disconnect can be resolved. So that is one of the other major benefits of having both Luminata and the Spectris platform together. So all those pieces come together in terms of the iControl tool itself. And for that, that's going to be the collaboration between Mettler-Toledo and ACDLabs that Joe DiMartino will be talking about next.
So everyone, right now, what I'll be discussing is that collaboration piece. And just to extend on what Matt was saying here, this is a slide that really represents what's occurring with this data-rich information. So if you're looking through this, a lot of users, they may start off with a design. It can start with within Chem ELN, maybe it's a design of experiment software. But once you get through that portion, you're going to plan that out through a lab reactor, in this case, iControl. Once planning out what you want to specifically look at for your settings, your parameters, you then reach the point of execution. And now when you go into the execution column of this slide, you're going to notice a lot of differing silos. You're collecting all the lab reactor information, which Matt mentioned is data rich and contains so much for temperature ramps and etc. But at the same time too, you'll have information from your LC, LCMS, and then from there, if you have an endpoint study as well, an NMR. And you'll note that all these are living in differing silos when it gets to analysis. And then from there, once you reach the point of decision making, that gets stored into an ELN as a PDF, MS Office, again, Excel documents. And that's where that knowledge is really tough to manage there. And what we've done with just going through and really understanding this type of workflow is using Luminata to really store and visualize all of that information together, viewing it from the lab reactor all the way from knowing that the Spectris processor can input from all these different analytical sources to have one place to really acknowledge what's occurring, seeing any types of trends. But since we can deal with any type of analytical data, in this case, like Matt mentioned, Waters or Agilent, we can make those type of decisions here as well. And this is what you're really going to view. And in a couple minutes, I'll be going into software and getting a better understanding and showing you what's occurring within ACDLabs. And what you're noting here is that what the software will do is when you're looking at time course data, it creates an in-process map, which is in the upper left-hand side. From there, it's using the knowledge obtained from the IC data center XML file and storing that as a live dynamic trend plot in the upper right-hand side. And then below that, you can also see a kinetic plot of getting a better understanding of the reaction, of where the reaction is completed, and just viewing the trends down the line, and also visualizing the analytical data. Again, things that are not easy to do, like I showed in that slide previously, that's all separated. And even if we wanted to look at them, they would be PDFs. And going a step further, if I was actually talking to Matt and going through that type of workflow, we'd literally be sitting through, looking at our ELNs, going through and understanding if he was looking at a specific stage and I was looking at something downstream, and we're trying to see what's been occurring. It takes a lot of time and effort. And what you'll notice once we get into the software, we try to minimize all of that information and store it all together. So this is the part of the live software demonstration. What I want to cover is the import of the XML file coming from iControl, associating any offline analytical data sets, and then from there, visualizing that data-rich information, viewing that dynamic and real live XML trend plot, viewing the in-process map, and comparing differing experiments. Now from there as well too, we can also update the kinetic plot. So I'll give you a brief background of what's occurring there. So right now you're looking at the software. And what the beauty of the software is, if you're new to Luminata, we're looking at one specific workflow, but there's many workflows that you can go and really envision and see down here as well. In this case, what I want to go through is if we're looking at this one-step reaction, we want to get the information from that lab reactor. Again, we decide to use a one-step in cases where it's 5, 6, 10 steps. You can also use the same iControl functionality here as well too. Now, just by selecting this intermediate or API product, I can then just say, okay, well, run iControl. And once I run iControl, you'll get this dialog box. And in this case, we can start saying, okay, well, what XML file do we want to associate to this project and to this process? So we have this XML file again from Mettler Toledo. And once we selected it, you'll note that right away, the software will do two things. It'll take all the numerical values coming in from the XML file and store it automatically within ACDLabs. But at the same time too, it will take all of the analytical data that was associated through the XML file. So just by putting in identifiers such as a name here, it can read all of these differing time points and place them together. Now, within this case, if it's already been processed, such as Matt mentioned, if you're using Empower, we can already take in that process data set. Now, if it's again coming from a different vendor, such as Agilent, and it has been processed yet, we can also take in that non-processed data set and we can associate an SDF with retention times to then run. So in this case, I could click OK, but just for simplicity and just in this webinar, I don't want anyone to just watch the ocean boil. So I just click cancel, and then we can just see the final set reaction inside of Luminata. So in this case, I'm just going to previously run reaction for this in-process map, and I changed the view so you can get a better understanding of what's occurring. And then from there, I'll select everything here too. And what you'll know, first thing, like I mentioned, it creates this in-process map. And this is, again, that one-step reaction that we're looking at, but it has all of the differing time points related to those sources of truth, like Matt mentioned. And we can see down the line where the reaction goes to completion. When I go to the dynamic trend plot, the reason why I want to just wipe everything from the screen is, again, this is coming from the XML. I want to show you the differing lines here that indicate the differing time points as well, so the user can see what's occurring. And what the beauty of the software is, you can start seeing that when I click anywhere here, excuse me, that you'll note that you can see those differing parameters that we're wanting to view down the line, getting an understanding of what's occurring. And also within this trend plot, the ability to associate any of the sources of truth, the data sets in here as well too. So just being able to view that and seeing that dynamically. Now down the line as well, if you're looking at the kinetic plot as well, these are taken from the in-process map and the analytical data. Within the in-process map, you'll note that you'll see the product and the starting material, and you can see the completion of that product as well. But like I mentioned, there's a couple of things that are quite interesting within the software. One of the greatest strengths of just using this piece of software is that we can illustrate and view all other runs that were made within XML and all the analytical data. So again, quickly, I'll just change my view so you can view it here. And I just wanted to show you one of our summary charts, or what we call a control chart here. And when you'll note within the control chart, it will show all the values from those different time points, from 18 minutes all the way to, let's say, 90 minutes. But what the beauty of the software allows you to do is really do an inventory of all of these types of reactions for this project and process to make a direct quantitative comparison. So all I have to do is click on a cell, and I tell the software, well, I want to check and summarize every project and process that's been looking at this one-step reaction. So in this case, I just say go into a setting called impurity master mode, and I go to all the runs that I've made. So in this case, I've done 3, and specifically, I don't have to look at every time point. I can say, well, I want to look at the end point, the last time point. I select it here, and once I do that and I click OK, you're going to note this table has changed. And really what I'm viewing here is this information from, let's say, a run that I've done. This is an ID identifier. But let's say down the line, a run that one of my colleagues did. So if Matt's done this run, and you'll note that in this record identifier, it's at about 92% of the product. This little piece that took 1 second, that could be a long conversation that we would have. Just like Matt mentioned, just imagine this piece where he's done his run, he's changed a specific parameter. I've also changed a specific parameter. I'm getting the area percents around 98%, and he's getting around 92. This takes up a lot of time. And once we reach this range, we don't have that time. In this case, like I've mentioned, this literally took us 1 second to just review this together. And again, now I don't have to search out and go on to his lab reactor, or ask him to connect to his ChemELN and find out more information, what's being run in the planning stage. Part of the execution within ACDLabs is the ability to view that, and that becomes a great strength, especially when you're looking at the lab reactor Mettler-Toledo piece. Now from there, I'm just going to go back to the previous view and just do one thing as well. You'll note that when looking at the data, maybe from the offline instruments, you've taken 5 or 6 points, but you may have taken something later on. Maybe you've taken our endpoint just to see what would happen if it reached 90 to 100 minutes. The software will allow you to still dynamically add data and time points to this in-process map and to the kinetic plots here as well too. So within the software, I can just say, well, I want to add an additional time point. Just by doing that, I can say, well, at 100 minutes, I want to add a new point, add endpoints. Then I can add the data file. Within the data file here, I can select it, and then just add stage. Just by doing that, you'll note right away in the background, once I've added the stage, it will add it within the in-process map, but dynamically also add it within the trend plot as well too. And you'll know in this case, it's already been processed. It adds this information directly, and it's been completed. And you'll see this new time point here as well, where if I zoom in further, you can see that additional time point. So really the beauty and strength of the software is taking this information and viewing this all together and being able to add additional information as well. And you'll note that now in the trend plot, you can see that down the line, it's been added here, and it's been added here. So to summarize, this is a data management challenge slide, and what we were trying to do with this slide, just like Matt emphasized earlier, even if we're in the same project team and we're looking at different steps, all users part of that team could be using different pieces within the organization. Some users could be using, let's say, any tools from Microsoft, such as Word, Excel, PowerPoint, and then storing that in SharePoint. Maybe we're using our ELNs, maybe we're using a CDS, or at the same time too, using something that's been built in-house. But as you can see within process and product development, this becomes quite a task, especially if you're looking through and using Excel or in Excel documentation to really store that knowledge and really try to identify what's occurring. What we've leveraged is the ability to store all of that together in one place and really make a central location for this type of data visualization, quantitative and qualitative analysis where we're in one place and we have a better understanding of what's occurring within all the analytical, rich analytical data that we have observed that is going on. If there's any questions, please feel free. Again, thank you for your time today. We really do appreciate it.